This video is a walk through the process of creating your membrane system in CHARMM-GUI. For those who don't know, CHARMM-GUI is basically like a Swiss knife which allows you to prepare input files for various types of simulations with different packages like GROMACS, NAMD, CHARMM, Amber, and the others. CHARMM-GUI is a collection of web tools, so you don't really need to install anything. You only need to configure your particular system, let it build it, and then you download it as an archive. As a CHARMM-GUI consists of different tools, there are multiple associated articles which you can cite if you find CHARMM-GUI useful in your research, and I strongly suggest you to do it. Another way to support CHARMM-GUI maintainers and developers is to donate them money. CHARMM-GUI can generate input files for various types of simulations, and you can explore the possibilities yourself by going to the input generator web page, and you will find links to CHARMM-GUI website and different pages in the description to this video. Today we are going to build a small membrane system for drug permeability studies. The dedicated tool is Membrane Builder, but if you are building something really large, you may want to consider using coarse-grained force field instead of CHARMM-36 force field. And CHARMM-GUI supports one of such force fields, Martini, and you can also build a membrane, but with a separate tool for Martini simulations. You first must decide if you want to build a protein membrane system or just a membrane-only system. And for the protein membrane system, you must provide a PDB file with structure information on your membrane protein, and then CHARMM-GUI will try to build a membrane around it. But in this particular video and for drug permeability studies, I will use membrane-only builder. The first step is the most crucial. You must decide the composition of each leaflet of your bilayer membrane and also the water thickness. And there are two ways to do each. As for the membrane composition, you can either provide the ratios, and CHARMM-GUI will decide for itself the exact number of lipids, or you can define the number of each lipid in your bilayer. As for the water thickness, you can also either provide the number of water molecules for each lipid, or you can provide the exact water thickness in angstroms. The membrane composition will depend on your study. If you are just studying the drug permeability for the first time, people also use just one stable lipid and in each leaflet. But in my case, I study bacterial membrane, in particular gram-positive membrane, and I have found the data in literature on the exact composition. I set the water thickness to 50 angstroms or 5 nanometers. I actually, in this particular project, I tried different thicknesses to achieve the best performance with no artifacts, but here this particular system is with 5 nanometers of solvent on each side of membrane. And after you have specified all the information, you just click on calculation of your system size. And then you can proceed to the next step. It is important that each leaflet of your membrane has roughly the same area in XY plane, and you will get a warning if it is not correct, if there is a large difference. And this is especially crucial for asymmetrical membranes because we have different lipid composition in each layer. The outer membrane of gram-negative bacteria, they have LPS, which is very large compared to phospholipids in the outer leaflet, and in the inner leaflet we have a basic membrane composition. You should also note that depending on how stable your composition is, CHARMM-GUI may fail in building membrane. It doesn't necessarily mean that your system cannot be reproduced, but it just sometimes it is just because of a wrong seed in building membrane, and you should just restart your building process with the same composition, and it may be okay or fail again. So it really depends on your luck, and also some membrane composition tend to be more stable than others. After your basic membrane was built, you can further proceed with adding solvent to your system and ions. But before that, you should, as it is recommended, to look at your membrane approximately. It is not yet built as it is in atomic detail, but you can also see the general layout of your bilayer. As for the ion placement, CHARMM-GUI allows you to provide the information on the concentration of ions, and you decide which salt to use to neutralize the negative or positive charge and produce physiological conditions. I recommend to use for most cases the default salt, which is potassium chloride. You can also use natrium chloride. In terms of physics, it is the replacement, but potassium chloride is more common. Certain lipids require divalent cations to be stabilized, like LPS that I have already mentioned, and it can be the calcium or magnesium as a combination, but in this case CHARMM-GUI sees that there is no such lipid, and you don't need them, so it does not allow you to select anything in this regard. But you can always add more ions if you need them. And after you are ready, you just proceed to the next step. At first, CHARMM-GUI builds the model of your membrane, then it builds components, and then it combines them together. At each stage, you can see the system at the current processing period. Here, in particular, is our membrane in atomistic detail. It is a small patch, but it should be enough for the kind of user intent. When you are ready, once again click to the button in the bottom right corner of your browser page, and you will proceed to the next step. The time required for each step, it depends both on complexity of your system and also on the current workload of the CHARMM-GUI servers, and it varies significantly. At each step, CHARMM-GUI performs some calculations in the background, and sometimes your system is caught in a loop, and the job fails due to timeout. In that case, you need to start over again, which is not very convenient. Also, I advise you to always save the ID assigned to your job, which is unique at each job, so you can retrieve the result of the job if you accidentally close the browser page, for example, or on the different machine. Step 5 is the last step where you can actually do some selections, which is the force field selection. It can be either CHARMM-36 or CHARMM-36M. For the membrane simulations, it doesn't matter because they only differ in CHARMM-46M has additional corrections of parameters for protein simulation, but we only model the membrane itself, so you don't really care. You can also also select amber first field if you need it. Then there is the selection of the package for which the input file should be provided, and there are multiple packages. I work with GROMACS. You can work with amber, CHARMM, or NAMD. Also, there are other force field related options like VYF, a parameter if you need to model cation P interactions more precise, but in that case you might also want to switch to Drude polarizable force field, which requires a different builder. Then hydrogen mass repartition is basically your hydrogen atoms, as far as I know, they are fused with heavy atoms, and this is if you want to switch from 2 femtosecond integration time integrator step to 4 femtoseconds, which means that your simulations will reach the desired time in 2 times faster, but the force field is optimized for 2 femtosecond steps, and you use links normally to account for oscillations of bonds to hydrogens. And I think for the most cases you should stick to the default without hydrogen mass repartition. And there is also multi-side calcium 2 plus parameterization, which is for the case when you study specific receptors where you need to, in more precision, analyze the dynamics of calcium ions and how they interact with proteins. CHARMM is famous for its 6-stage equilibration. It provides all the required files for energy minimization, sometimes 2-step energy minimization, and also equilibration with first NVT and then NPT ensemble, and also the production run MDP file. Obviously, you will need to edit them for your case, but they're also fine on their own. They should not contain any errors. You can proceed with them directly. As for the temperature, I here select 301.15 kelvins, which is 28 degrees Celsius, and it is not the default temperature, it is slightly lower. And this is because I wanted to match the experiment's temperature, and I work with soil bacteria, whereas human pathogens, they usually are okay at higher temperatures, and this is why most of the CLSI standards for MIC determinations, they use 35 plus minus 2 degrees as their common temperature. Well, it really depends on what you study. But in the case of membrane simulations, it is also crucial that your lipids have a low enough melting point so that at lower temperatures they do not transition into the solid phase, because then obviously your drug cannot transition through membrane. But then again, if at the experimental temperature it cannot transition, then your simulation should match your experiment. Finally, you can download your input files. Before we proceed to the more detailed visualization, let's check the folder of the downloaded archive. README file contains general info, but it can also be adapted into a script to automatize the process of your system minimization, equilibration, and starting the production run. We have multiple MDP files for energy minimization, equilibration, and in the end the production run. Equilibration files, they differ in position restraints, and In all cases, we use a combination of VD-scale thermostat and series-scale barostat to reach the reference temperature and pressure, and to also maintain it during the production run. The choice of thermostat, barostat, and their combination, it really depends on what you simulate and what are your preferences. In textbooks, you can also find the combination of Nose-Hoover thermostat and Parinella-Rahman barostat as a canonical combination of methods for temperature pressure control. But nowadays we shift towards more robust methods like the ones that are in the basis of VD-scale and series-scale thermostats and barostats, and you can also use their combinations like VD-scale for temperature, Parinella-Rahman for pressure. But the limitation of Parinella-Rahman is that it cannot be used for poorly equilibrated systems. So you, in any case, during equilibration, you need to reach the reference pressure using series scale, barostat preferably. And then you can switch to Parinella-Rahman. And the same comes for Nose-Hoover. It is best for maintaining temperature, but not for reaching it. And your system can just blow up if you start with Nose-Hoover and Parinella-Rahman. But you can either switch between them, or you can use VD-scale and series scale, and it will be fine for both equilibration and production run. You may also notice that we have semi-isotropic pressure coupling instead of just isotropic. That's because it is specific to membrane simulations. And the limitation of series scale barostat is that it cannot currently normally reproduce anisotropic pressure coupling. For that, you should use Parinella-Rahman. Another thing you may notice by observing your equilibration files is that at early stages of equilibration, CHARMM-GUI often sets your time integrator to 1 femtosecond step instead of 2 femtosecond, which is normal for CHARMM-36. That's because it wants to capture even slightest vibrations of bones in your system at the beginning, and later you switch to 2 femtosecond step. But don't be surprised if the early stages take like twice as much time for the same trajectory interval as later stages. Finally, we can make a better visualization of our system. I will use for that ChimeraX, but you can use any system for molecular visualization which can handle large system like PyMol or Maestro Schrödinger or VMD, if you like. You should know though that most of the existing software doesn't have a specific cartoon or ribbon tube representation for lipids, and you usually just use a surface to show the membrane. But ChimeraX also has worms. You can kind of adapt them worms representation for membrane, but I won't show this in this particular video. I don't think that it is necessary really. And this is pretty much it for today.